I'm Marjan de Mey. I'm professor at Ghent University, and my group is mainly focusing on developing bioprocesses using microbes as a cell factory.
I'm Pieter Coussement. I work at ML6, an AI/ML consultancy company, and I'm a senior data engineer and squad lead at the company.
Plant metabolites are very interesting molecules that are produced by plants and that have a whole bunch of bioactivities which are very interesting for us. It could go from flavors like stevia. It could be also having more medical application like an anti-cancer treatment. Today, these products are mostly extracted from plants, which is not always that easy. Because the plant is not producing that molecule in huge amounts, and so it's not really economically viable, but it's also not sustainable. So in DeCYPher, we really want to see how we can produce these molecules in a sustainable way and in a good way.
We're altering nature, in a sense, the bacteria, to produce a high-value molecule, but are we going to spread it out outside? No, we're going to use it in a metal vessel where it's going to produce the molecule we want, and afterwards everything is killed off and it does not come into nature.
It's actually an enzyme that is attached in a membrane in the plant, and that one will do what we call a biochemical reaction called oxygenation. So easily said, if you have a plant scaffold, it will add an OH group on one of the points on the scaffold. And that's what we call the decoration. Typically, these SIPs are very specific, and so you have one specific SIP for one specific decoration on the scaffold. So that's the search that we have to do. We want to find and learn how this enzyme looks and how it reacts, or what the features are of this enzyme, and to match it to the specific decoration. So the number that we are targeting within DeCYPher is huge because there are many plant metabolites, and if we look at terpenoids and flavonoids, then we are talking about 10,000 to 20,000 different compounds for each class that we can reach, which is important if we want to really gain the advantages of these molecules, because they can have a huge impact on our daily life.
I think biology is the perfect use case for machine learning, because it actually is able to catch relations between ins and outs that are very hard. That's the core of machine learning, I think, to capture those. But that's also exactly biology. There is, at this point, a certain unpredictability in biology, whether it's a microorganism, a human, or whatever. There is I call it the biofactor, unpredictability. And that unpredictability is just the core of machine learning.
In this project, we really want to merge synthetic biology and machine learning or active learning. And we typically will do this in the design, build, test, learn cycle. This is a cycle that synthetic biologists use as an approach all the time. And in these 4 steps, especially in the learning step and the design step, there is where we want to bring in AI. So it's not like that in the lab robots will be there. It's mainly the wet lab people who will do experiments, and the data that they will generate will feed the models for the learning. And as such, these models will then steer us again to say, look, this is the next experiment that you have to do to learn better and to get a better, improved microorganism.
I think synthetic biology as an engineering field is difficult. There are a lot of things we don't yet understand on biology. That's one side. On the other side, biotechnology will have an important impact in our future. And I think with AI, also a field evolving very fast, we can really capture the unknowns, the unknowns even, of biology to help and drive biotechnology forward.